Welcome to Unacademy EAMCET YouTube channel, students. So once again, good evening all of you students. Welcome to Unacademy EAMCET YouTube channel. So Unacademy EAMCET YouTube channel, on Unacademy EAMCET YouTube channel, every day at 4 p.m. we'll be having the biology class, students. So as usual, today also we have biology class that is starting now, that is on the topic that is biotechnology. So we'll be discussing about the topic that is called as the biotechnology. So it is the most advanced branch of biology nowadays. So it is very helpful to all the human beings, this particular branch of biology, that is biotechnology. So before starting this lesson, I would like to introduce about myself. Before starting this lesson, students. So before starting this lesson, I would like to introduce about myself. So myself, Dr. Rajendra Prasad. This is myself. My name is Dr. Rajendra Prasad, and my qualification. So if you see, this is my qualification. I have completed PhD in biological sciences, and I'm also UGC NET qualified in the life science subject. So I have been teaching biology. So I have been teaching biology to NEET medical aspirants from last 13 years. So I have taken coaching classes for NEET medical entrance examinations and AAPMT, AIMS, JIPMER, KCET and EAMCET. So from last 13 years I have been teaching the biology to NEET medical aspirants of different categories like NEET and as well as AAPMT, AIMS, JIPMER, KCET and as well as EAMCET. So I also worked with the institutes. So I also worked with the renowned institutes like Sri Chaitanya Narayana Education Institutes, and I have mentored several thousands of students. So I have mentored several thousands of students to achieve their goal as a medicos. So this is about myself. Students. So before starting our Unacademy EAMCET YouTube class, so I would like to introduce about our Unacademy Plus classes. So Unacademy is providing daily free live classes on its YouTube channel. Apart from that, Unacademy is also providing special live classes on its app. So apart from special live classes, there are paid classes. Those are called as Unacademy Plus classes. So what is importance of this Unacademy Plus classes is a designed course for complete medical entrance examination preparation will be is prepared. So that is the reason why this particular plus subscription is very important actually. So a designed course. So there is one batch was already started, that is called as a comprehensive batch. So there is a batch was started. The name of the batch is called as comprehensive batch. This batch was started in the September month. So in that comprehensive batch, I will be taking the biology classes, and Sir Durga Prasad sir will be taking the physics classes, and Sachinarayan sir will be taking the chemistry classes. So what is importance of this particular batch is, so this batch will run up to end of the February month, and by end of the February month we will be completing the intermediate first year syllabus and as well as intermediate second year syllabus, so which is required for the coming medical entrance examination of 2021 EAMCET. So the respective educators will give tips and tricks also to solve the numericals, especially in case of physics and chemistry, and topic wise practice papers will be provided, and a full-length mock test. So full-length mock test also will be conducted. So these all, so if you take subscription, you can avail all these benefits. So if you are interested, you can use my code Rajendra Live. So if you use my code Rajendra Live, so you will get all these benefits. So you can take 1 month plan. So you can take 1 month plan. So it is of 2,250 rupees. If you use my code Rajendra Live, you will get the 1 month plan for 2,250 rupees actually here. Okay. So Unacademy is not only 1 month. Unacademy is providing the long-term plans. So that long-term plans is like a 6 months plan and 12 months plan. So 6 months plan. So 6 months plan is of about 10,125 rupees, whereas 12 months plan. So 12 months plan is about 12,375 rupees. So this is the best plan, students. I suggest you to take the 12 months plan. So complete preparation will be there. So course will be completed by end of the February month in our batch course. So after that you can start practicing the multiple choice questions on our platform. So there are many lecturers in all subjects. You can attend those classes and you can prepare you can attend several mock tests will be conducted. So I suggest you to take the 12 months plan. So if you are interested, you can use my code Rajendra Live so that you can get the 6 months plan for about 10,125 rupees, whereas 12 months plan you will get for 12,375 rupees. Then what is importance of this plus subscription is a premium content. So premium content will be available at affordable price, and a structured live course and dedicated live doubt clearing sessions will be conducted after every 3 classes. After every 3 teaching classes, fourth teaching class will be a doubt clearing session. So you can interact with the lecturer and you can clear your doubts actually, or you can interact with the educator in the respective subjects, and quiz will be conducted at regular intervals, and several batches. So batches will be prepared, so you can attend the batch which you are interested, and test series. So test series also will be available on our website, students. So this all you can get it for a single subscription of 12 months plan of 12,375 rupees and 6 months plan of 10,125 rupees. So if you are interested, you can use my code Rajendra Live so that you can get the course for 12 months plan of 12,375 rupees and 6 months plan of the 10,125 rupees. So that is about our Unacademy Plus subscription, students. So welcome to Unacademy EAMCET YouTube channel, students. So welcome to students. So welcome to Madhurima, welcome to Komala, welcome to Sai Deepika, so welcome to Sai, so welcome to Shiva, welcome to Harish, welcome to Usha, welcome to Nishita, so welcome to Balu, and welcome to Priya. All of you, welcome to Unacademy EAMCET YouTube channel, students. So once again, all of you, so good evening all of you, students. So in today's class we'll be discussing the lesson that is about the biotechnology. We'll be discussing about the biotechnology topic in today's class. So welcome to all of you, students. So welcome to Unacademy EAMCET YouTube channel. You subscribe our EAMCET YouTube channel, students, so that you will know the timings of the particular lectures that is going every day. So every day daily we have 4 p.m. biology class. We'll be having 4 p.m. biology class, so that you'll be knowing what topic is going to take class in tomorrow's class. So like that you will be knowing. So అందరూ subscribe చేసుకోండి. So if you subscribe, you will come to know the timings of the subjects, and topics also will be coming across. Okay, so let us start the lesson, students. So welcome to few more students. So welcome to Jayant, so welcome to Aarti, so welcome to Unacademy EAMCET YouTube channel, students. Welcome to Aarti, welcome to Jayant, so welcome to Unacademy EAMCET YouTube channel. So let us start our lesson, students. So let us start with the first, so biotechnology. So no doubt this biotechnology. So no doubt biotechnology is it is a branch of biology. So it is a branch of biology. So it is advanced branch of biology actually. Okay. So this involves integration. So this integration means it is a mixture of several. It is a mixture actually. Biotechnology is a mixture of several advanced courses or advanced branches. So that's what we are showing here. So biotechnology is an integrated branch. No doubt it is an integrated branch. So it is an integrated branch of biology. So which involves, which also involves the biochemistry, so which involves microbiology, genetics, cell and molecular biology, immunology, environmental sciences, and engineering sciences. So this biotechnology is an integrated branch. Okay, so it is an integrated branch. So the the basics of biochemistry was also part of biotechnology. The applications of microbiology are also part of biotechnology. So the genetics part of the plants, genetic part of the animals, so they are also part of the biotechnology. So molecular biology like DNA replication, transcription, translation, they are also part of the biotechnology. And immunological aspects, antigen antibody reactions, nature of the antigens, nature of the antibodies, so these all are part of the biotechnology. So that is the reason. So how the biotechnology branch was defined, it is an integrated branch of biology. So it is an integrated branch of biology. It is an integrated branch of biology. It is integrated with other branches. What are the other branches which it is integrated? It is integrated with biochemistry, microbiology, genetics, cell and molecular biology, immunology, environmental sciences, and engineering science. So these all branches are integrated. So let us see that is the applications of this particular biotechnology. So what happens in biotechnology? It is an application. So what is a biotechnology? No doubt it is an applied branch of biology. So in this applied branch of biology, what happens? It is an application of techniques. So no doubt it is an application of techniques using the live organisms. So in this biotechnology, so what happens is we are using, it is no doubt it is a technique. Okay, it is no doubt it is involved the techniques. In this techniques, what happens? We will be using the live organisms. So live organisms means those organisms which are living. So we will use the living organisms to get our desired product, and those desired products are essential for human life day to day. Okay, such is called as a biotechnology. So biotechnology involves the application of techniques in which the living organisms are used to produce the products which are essential for the human beings. Okay, so biotechnology involves, so what biotechnology actually involves? It includes recombinant DNA technology, gene cloning, and as well as gene therapy. So you have to remember these three terms. In multiple choice questions they'll ask you, so what is this biotechnology includes? So biotechnology includes, no doubt it includes recombinant DNA, it includes gene cloning as well as gene therapy. So these are part of the biotechnology actually, students. Okay, so welcome to few more students. So welcome to Aman, welcome to Saurabh, welcome to More to Learn. So welcome to Unacademy EAMCET YouTube channel, students. So welcome to all of you once again. Good evening. Okay, so before starting this lesson, I would like to discuss about the term that is called as a fermentation. Okay, so fermentation, so from age old practice we are doing fermentation. Okay, so fermentation is the oldest biotechnology process. This is a very important point you have to remember here. So which is the oldest biotechnology process? It is nothing but fermentation. So in one of our lesson in 12th class, one of our lesson, there is one lesson named called microbes in human welfare. So in that microbes in human welfare, we have seen about the curd preparation we have seen. So the curd preparation is, so no doubt curd preparation is, no doubt it is a fermentation process. No doubt curd preparation is a fermentation process. Similarly, the preparation of cheese. So in preparation of cheese also we are using the living organisms. So similarly, the preparation of the organic acids. So preparation of organic acids like citric acid, butyric acid, acetic acid, and preparation of antibiotics. And similarly, preparation of the beverages. So these all nothing but this involves this is also an application of biotechnology only. So which is the oldest biotechnology process? So like that they'll ask in multiple choice question, which is the oldest biotechnology process? Then we have to clearly remember that the oldest biotechnology process involves nothing but the fermentation. So in the olden days we don't know that we will be doing fermentation. Like in our household activities, we prepare curd by taking the starter culture of the curd from the previous day. Okay, so we don't know that it is also a biotechnology process. So that is the reason the oldest. So in the preparation of the curd, what happens? It is nothing but fermentation process, and fermentation takes place with help of the microorganisms. So in olden days also biotechnology process took place, and it is by a process that is called as a fermentation. So that is the reason the fermentation is also known as the oldest biotechnology process. So by using the principal fermentation, the curd preparation takes place, the cheese preparation takes place, the synthesis of organic acids like citric acid, acetic acid, butyric acid. So these all are important for humans in day-to-day life. So these all acids are prepared. Similarly, antibiotics are prepared. So antibiotics are extracted from the organisms and they are used for the human beings, for human welfare. Similarly, beverages, especially the fermentation part. Fermentation means we have to tell about beverages only. We in preparation of the beer industry, okay, and a wine and different other rum, all these. So vodka, this all involves the preparation of beverages. So beverage industry is totally depending upon the fermentation only. So that is the reason. So fermentation is considered as a oldest biotechnology process. So now, so which is the modern? So that is the oldest. So oldest biotechnology process involves the fermentation. So modern biotechnology process. So modern biotechnology process. using the genetically modified organisms. So modern biotechnology process, what they are using, so modern biotechnology process are using the genetically modified organisms. Okay, so these genetically modified organisms are named as like GMO. So in our textbook they will write as GMO crops. So like GMO crops, like that, right? So GMO means it is genetically modified crops. So modern biotechnology involves the usage. So usage of the crops, genetically modified crops, genetically modified organisms are used for the crops. Okay, so how they are prepared? So by altering, so how they are prepared? So genetically modified organisms, how they are prepared? So how they are prepared? They are prepared by altering the chemistry of DNA and constructing the recombinant DNA. So finally what we have to tell here is, in modern biotechnology, so genetically modified organisms was possible. So genetically modification of organisms is taking place. And how we are genetically modifying the organisms? We are modifying the organisms by altering the chemistry of the particular organism. Okay, so that altering of the chemistry of the DNA, such process is called as a recombinant DNA. Such process is called as a recombinant DNA technology. So such process is called as a recombinant DNA technology. So what is the recombinant DNA technology? If you see here, is it is nothing but altering. So it is altering the structure of the DNA and producing the modified organism, and that modified organism will be helpful for the human welfare. Such process is called as a recombinant DNA technology. So that is about the recombinant DNA technology definition, students. So welcome to few more students. So welcome to Aarti. Yes, Aarti, you are telling, right? So lactobacillus. So lactobacillus is involved in the preparation of the lactic acid, no doubt about it. Similarly, it is also involved in the process of the preparation of the curd. Okay, so very good. So similarly, so Begum was asking to take EAMCET practice questions. Actually, EAMCET practice questions on our app. So on our app, our Unacademy EAMCET YouTube channel, I have taken nearly 15 grand tests. Okay, already recorded sessions were also there. If you are interested, you can go through those sessions, students. Nearly 15 grand tests I have taken from last 1 month. So you can just go through those. So yes. Okay, so anyway, we will be discussing in depth only this biotechnology lesson. Okay, so welcome to few more students. So welcome to Komala. So welcome to Komala, students. Okay, let's continue, students. Okay, so finally what is this recombinant DNA technology? So what is this recombinant DNA technology? Is nothing but it is altering the chemical structure of the DNA. Okay, altering the chemical structure of DNA will produce a genetically modified organism. So that genetically modified organism will be certainly helpful for the human beings in day-to-day life. So that is what called as a recombinant DNA technology. Okay, now let us see about the overall process. So overall process, what is happening in biotechnology? So we will be seeing overall process, what are the steps are involved in the process of the biotechnology. So this is a very important diagram, students. You should understand this. If you understand this, certainly this lesson will be the easiest lesson for you. Okay, so let us see the diagram carefully. Let me explain. Yes, 1 minute, students. So Madhurima was asking me one question: how to connect the special classes, students? So how to connect special classes of Unacademy EAMCET means you download the Unacademy EAMCET. So you first to download Unacademy Learning app. Okay, you download Unacademy Learning app, students. After downloading Unacademy Learning app, okay, in the course you type EAMCET. Okay, in the course, your course name will be there. You type there EAMCET. So when you type the EAMCET, then what happens is they will show the educators which are present in the EAMCET platform. So there are nearly 35 educators were there. So you can attend those people's classes. You can check my class also, Rajendra Prasad, Vavilpalli, my name is. You can type in Unacademy Learning app, my name Rajendra Prasad, Vavilpalli. So just now I shown you my details in the very starting of this session. You can type my name directly on Unacademy Learning app so that you can directly connect to my classes. Okay, So Madhurima, so that is how you can connect. So download Unacademy Learning app. After downloading Unacademy Learning app, type the course as EAMCET. Okay, in that EAMCET course you can find the lecturers and you can go through the topics which you are interested. Okay, let's continue with this topic, students. So what are the steps in the biotechnology if you see? So first, what is the step in the steps? Okay, so let us take, for example, I am taking one example for preparation. For example, I am taking one example like preparation of, okay, for example, I am taking, students, one preparation of insulin. Okay, I am taking one example to make you understand. So preparation of insulin. So what I am explaining here is I am taking one example of insulin. Okay, so how to prepare insulin in the laboratory, okay, in a large scale, how to prepare insulin. So how to prepare insulin means, so no doubt for insulin gene, so insulin gene is present on the humans. So insulin gene is present. Okay, we have to first, what we have to do is we have to separate the insulin gene. Okay, so what we have to do, we have to separate the insulin gene from the human DNA. So where insulin gene is present, insulin gene is present on human DNA. From human DNA we have to separate the insulin gene. Then for that, what we will do is, so for example, now this is the DNA, for example, this is our human DNA. Okay, take this as a human DNA. So what is your purpose? So our purpose is to separate the insulin gene. Okay, insulin gene is present on the human DNA. For example, our insulin gene is present somewhere here. Now we want to extract this. Okay, so this is the human DNA. From this DNA, first we want to produce insulin in a large amount, in a large scale. So for that we are using recombinant DNA technology. So let us now discuss the steps in the recombinant DNA technology and steps in the biotechnology. Okay, so we are taking one example to extract insulin. Now this is the human DNA. Think this is a human DNA. On that human DNA, here is our insulin gene. Now this insulin gene containing DNA is treated with RE. What is this RE means? Restriction enzymes. So when you treat with restriction enzymes, what happens? Restriction enzymes will cut at specific region. Okay, now this one, now these are the fragments. Now this is our fragment. What is our fragment? Now this fragment is our insulin gene. Okay, this fragment is our insulin gene. Okay, now this gene is our gene of our interest. Now all these blue fragments. Okay, so in these blue fragments you can see this is the gene of our interest. These are the insulin genes. Okay, so this is our gene of our interest. Okay, so yes, so once again, students, so what I am explaining you is how to produce insulin, okay, in the industry in a large amount. Okay, so how to produce insulin in industry in a large amount? First step is, what we have to do is we have to extract the human DNA first. So we have to isolate the human DNA from the human cell. After isolating the human DNA, in that human DNA we have to exactly identify where is the insulin gene. Okay, and that insulin gene we have to separate. How to separate the insulin gene? We have to use restriction enzymes. So now this is our human DNA and we are adding restriction enzymes. When you add restriction enzymes, what happens is our insulin gene, this is our insulin gene, our insulin gene is separated. Okay, now that insulin gene is attached to a vector. Now you can see here, so you can see here, this is the vector. So vector is used. Generally the vector used is it is nothing but bacterial plasmid. So bacterial plasmid is used as a vector. Okay, so bacterial plasmid is used as a vector. Okay, now what happens? So this all process, whatever process which we are seeing is all this takes place in a test tubes. Okay, it is not taking place outside. Now what happens? Now this is the vector. So the vector and insulin gene, so these two are treated. Then what happens? They get, so now what happened? Our insulin gene, this is our insulin gene, that insulin gene is attached to the vector. So insulin gene is attached to the vector. Okay, so now this is this molecules where the gene of our interest, our gene of our interest is attached to the plasmid. Our gene of our interest is attached to the plasmid. that molecules are called as a recombinant DNA molecules. Okay, those are called as a recombinant DNA molecules. Okay, so after attachment, what happens? We have to, so this plasmid, this one recombinant DNA molecule has to be transferred into a bacteria. Now see, in this next step what happens? It was transferred into the bacteria, and this process of transferring the recombinant DNA molecule into bacteria, such process is called as a transformation. So such process is called as a transformation. So what happens here? The recombinant DNA molecule, okay, so what is this recombinant DNA molecule is composed of? It is composed of our gene. What is our gene? Our gene is insulin gene. Okay, it is a combination of insulin gene plus vector. It is a combination of insulin gene plus vector. Because of that reason, it is called as a recombinant. Okay, so recombinant means it is a combination of two different things, is called as recombinant. So what is that recombinant is composed of? It contains insulin gene and vector. Okay, so that recombinant, okay, the recombinant DNA cannot multiply outside, so it has to be transformed into a host. Now which is the host used here? Now this is the host. This host is nothing but, this is the host. The host is nothing but, it is the generally host used is E. coli. So E. coli is used as a host actually. So E. coli is used as a host. So host is generally is E. coli. E. coli is used as a host actually. Okay, so can anybody tell me why E. coli is acting as a host, students? Anybody? Can anybody tell me why E. coli is acting as a host? Yes? Anybody, students? So why anybody can tell me why E. coli is acting as a host? That is my question. Why E. coli is, why we are taking E. coli as a host? Yes? Anybody, students? Anybody can answer, please? Yes, students. Yes, friends, anybody? Yes. So why E. coli is used as a host? That is my question actually. Why E. coli is acting as a host? Okay. The reason is because, so Aarti was telling that it is affects fastly, but actually why E. coli is acting as host? Reason is because, so E. coli duplicates for every 20 minutes. Yes, very good, Komala. So it replicates for every 20 minutes. It duplicates. Okay, it duplicates faster. It duplicates or replicates. It duplicates or replicates. Okay, duplicates or replicates. Okay, it duplicates or replicates for every 20 minutes. For every 20 minutes, it keep on replicating or duplicating. What happens is, for every 20 minutes, it keep on replicating or duplicating. So that is the reason why what happens is E. coli is generally used. Okay. Yes. Yes. Welcome to few more students. Welcome to Rahmat. Yes. So what is the, why E. coli is generally used as a host? That is my question, students. Why E. coli is used as a host is because E. coli duplicates faster or replicates faster. So for every 20 minutes it undergoes binary fission for every 20 minutes. Okay, so that is the reason E. coli is taken as a host actually. Okay, so now what happens here? So that is the reason E. coli is taken as a host. So in this diagram, if you see clearly, okay, so bacteria, okay, so bacteria E. coli, so bacteria E. coli is used as a host. So inside the bacteria, what happens is the recombinant DNA molecule is transformed into the host, that is E. coli. So that transformation, of this particular recombinant DNA molecule into the E. coli, so that process is called as a transformation. So inside E. coli, now what happens? So inside E. coli, so this recombinant plasmid or recombinant DNA molecule can also be called as a recombinant plasmid. So this recombinant DNA molecule starts replicating and produce multiple copies. Such type of process is called as a cloning. Now you can see here. So initially we have only one recombinant DNA molecule. After that, what happens? Replication took place and it produces two recombinant DNA molecules. Later again, so now this process, so like this, Now this process is called as a cloning. So now this process is called as a cloning. So multiple copies. So multiple copies. So what is this process of cloning means? It is the production of multiple copies. So multiple copies will be produced. So multiple copies of the recombinant DNA molecules will be produced inside the bacteria. So very fastly. Very fastly means how much fast? For every 20 minutes the bacteria undergoes binary fission and it produces multiple copies. Okay. So like this what happens? So these all process. So where is this all process is taking place? If you see this all process, like this all process will be taking place inside a bioreactor, that is called as a fermenter. This all process takes place in the fermenter. Okay. So this all process will be taking place in the fermenter, so that we will get the multiple copies of the insulin gene. So we will be getting the multiple copies of the insulin gene. After getting multiple copies of insulin gene, then expression of the insulin gene will be taking place. So what is the meaning of this term expression? Means the insulin gene, the recombinant insulin DNA. Okay. So recombinant insulin. Okay. The recombinant insulin DNA. Okay. Recombinant DNA. Okay. Recombinant insulin DNA will express. So what is the meaning of this term express? So what is the meaning of this term express? Means it will undergo transcription. It will undergo transcription to produce mRNA. Okay. And this mRNA will produce protein. This protein is called as a insulin protein. Okay. This protein is called as a insulin protein. Now this insulin will be extracted and it is purified and it is used for the human welfare. Okay. So once again I will explain, students, once again I will explain this process. So yes students, some students were asking me one question. So Harish was asking me one question. So bacteria replicates very slow actually. So he's asking one question. So why bacteria replicates very slowly? But bacteria does not replicate slowly. For every 20 minutes it keep on duplicating. So for every 20 minutes it keep on duplicating. It replicates fast only. Okay. So during the process of replication what happens? The DNA has to separate. Okay. So the binary fission. So binary fission is taking place in bacteria. That binary fission is the amitosis. It is taking place very fast. The binary fission is not mitosis or meiosis. It is amitosis. Okay. For every 20 minutes it keep on dividing. Okay. It's very divides very fast. Okay. Then there is one more question was asking. Yes. So Komala was asking me one question. So bacteria undergoes fission or binary fission. So actually the term binary fission is the full complete term. So binary fission, it undergoes binary fission. Okay. So fission is nothing but it is a binary fission only. In some books they write as a fission, but it is binary, means when fission took place it produces two identical daughter cells. Such type of fission is called as a binary fission. Bi means two, so bi and two kada. So bi means two. Okay. When fission is taking place, when separation is taking place, it is producing two daughter individuals. When two daughter individuals are producing through fission, such type of fission is called as a binary fission. If several daughter cells are produced through fission, it is called multiple fission. Okay. Fission can be bi or multiple. Okay. What is binary fission? During the fission only two daughter cells will be produced. Such condition is called as a binary fission. So during fission if multiple daughter cells are produced, such condition is called as multiple fission. So like that there is a difference between the binary fission and multiple fission. Okay. So like that we have to remember, students. Okay. Anyhow, so welcome to few more students. Welcome to Shashidhar. So welcome to Shashidhar. Okay. So once again I will explain you students clearly this what are the steps in biotechnology if you see. So I am taking one example. So I am taking one example to explain you how the steps in biotechnology is taking place. Okay. So I am taking one example. So what is that example is? I have taken one example like, so we want to prepare insulin. Okay. We want to prepare insulin. Okay. We want to prepare insulin protein in the laboratory or in industry. How to prepare that insulin protein, that is the thing. So there are some steps are there. So that steps are nothing but by using biotechnology process we will prepare the insulin protein. So what are the biotechnology process? What is the first step? So insulin. So to produce insulin protein we should identify the insulin gene. So where is that insulin gene is present? We know that insulin gene is present in our human beings. So we have to extract the DNA of human beings. Now first we will extract the DNA of human beings or isolate the DNA of human beings. Okay, to that isolated DNA we'll add restriction enzymes. When you add restriction enzymes, we should add restriction enzymes, those are specific. Okay, those are specific and separates only insulin gene on the human DNA. So like that, so we are thinking that blue color one, this one. So this blue color one is our human DNA, and we have added our restriction enzymes so that it is cutting the gene, that insulin gene, that gene is called insulin gene. So like that, multiple copies of insulin genes I am showing here. Okay, 4 insulin gene copies. So now that insulin genes are now inserted. Now insulin genes are inserted into a vector. Okay, so this is the vector. What is source of vector? If you see the source of vector, generally the source of vector is bacteria plasmid. Bacteria plasmid is the source of vector. Okay, we will discuss in detail in the coming class about the properties of vector, but the source of the vector is bacteria source or plasmid. Okay, now what we will do, we will take this insulin gene and we will fuse with the vector. So these particular genes, when we cutting with restriction enzymes, it has one overhangings. These are called as sticky ends. So sticky ends will be created. So if sticky ends are not present, what happens is it cannot easily attach to the vector. So we will choose those restriction enzymes, okay, which will create sticky ends, okay, so that the gene of our interest, which is insulin gene, can easily insert into the vector. So the same thing happened. So finally insertion took place. So here you can see 3 successful, okay, 3 successful insertions, and those are called as a recombinant DNA molecule, and those are called as a recombinant DNA molecules. Here you can see 3 recombinant DNA molecules were shown in the pictures. Now this recombinant DNA molecules cannot duplicate or replicate by itself, so it should require some host. And what is the host we have chosen? We have chosen E. coli bacteria, okay, Escherichia coli bacteria as a host. Now the question arises like, why Escherichia coli is taken as a host? Why only Escherichia coli is chosen as a host? There are several millions of bacteria. Why Escherichia coli is taken as a host means Escherichia coli or E. coli is the only bacteria which can duplicate or replicate for every 20 minutes. It keep on replicating or duplicating for every 20 minutes. So that is the reason we have taken the Escherichia coli bacteria. Okay, so that is the reason. So that now the transfer of recombinant DNA molecule. So transfer of recombinant DNA molecule into the host, and that process is called as a transformation. Now what happens? The recombinant DNA molecule now entered into E. coli, and inside the E. coli cytoplasm our recombinant molecule is present. You can see our recombinant molecule is present here inside the cytoplasm of E. coli, and the recombinant molecule, okay, will start replicating. So you can see that, and it starts replicating and produce another identical recombinant DNA molecule. Such process is called as a cloning. Okay, so like that, numerical copies of the, okay, several copies of the recombinant DNA molecules will be produced. So if recombinant DNA వచ్చింది కదా, దాని నుంచి మనకి ఇన్సులిన్ ప్రోటీన్ కావాలి. So then what happens? So the expression of the recombinant DNA molecule takes place. ఇక్కడ ఏంటి expression అంటే ఏంటి? So what is the meaning of that expression means? ఇక్కడ చూసారు కదా, recombinant DNA. So the recombinant DNA which is having the insulin gene, the recombinant DNA will undergo transcription. ఎప్పుడైతే transcription జరిగిందో, it will convert into messenger RNA, and that messenger RNA will produce the protein. So such process is called expression. So term expression, whenever they are using term expression means we have to identify it as like transcription is taking place and producing mRNA, mRNA is producing protein through a process that is called translation. So transcription, translation is taking place. Okay, altogether is called as a expression. దాన్ని expression అని చెప్తారు అన్నమాట. Okay, so like that, these are the steps in case of the, so these are steps in the biotechnology. In detail, one step we will discuss actually. So final step which we are seeing here. So final step ఇక్కడ చూసాం కదా, this steps, all these steps. So all these steps takes place inside a bioreactor. This all steps takes place in a bioreactor. So that bioreactor is nothing but it is called as a fermenter. So it is called as a fermenter. This all process takes place inside a bioreactor, or it is a fermenter. Okay, so welcome to few more students. So welcome to Hafiz. So good evening students, once again good evening all of you. So welcome to Hafiz. So welcome to Karna also. Welcome to Karna. Good evening once again, all of you. Welcome to Karna. Let's continue, students. Yeah, welcome to Asmita. So welcome to Asmita also. So welcome to Unacademy EAMCET YouTube channel, students. You subscribe our EAMCET YouTube channel, students, so that you can get the details of the timings of the class. Okay, so let us continue. So let us see. So there are two main techniques. So there are two main techniques which are used in the biotechnology. So there are two main techniques which are used in biotechnology. Just now we have seen the oldest application of biotechnology is nothing but fermentation. Okay, then at the same time we have seen the recent advanced technology of biotechnology involves the recombinant DNA technology. Similarly, the two main techniques. So two main techniques generally used in biotechnology is nothing but it is the genetic engineering. Okay, and next one is biochemical engineering. So these are the two techniques. So these are the two main techniques that is used in biotechnology is so genetic engineering and biochemical engineering. So biochemical engineering and genetic engineering are the two main techniques generally used in the biotechnology. Okay, so in genetic engineering, what we are doing in genetic engineering? So genetic engineering involves nothing but manipulation. So we are manipulating the DNA, So we are manipulating the DNA. So in biochemical engineering, so biochemical engineering involves nothing but fermenters, okay, or bioreactors. Fermenter ki inko పేరు ఏంటంటే bioreactors. So fermenter and biotech growing of the organisms. So growing of the organisms అంటే E. coli. So E. coli ni will be growing in a huge quantities in a large fermenter or bioreactor. So that process of growing the E. coli, okay, that actually E. coli is acting as a host. Inside E. coli only our recombinant DNA molecule is present. So growing of these bacteria or growing of these microbes, okay, growing of these microbes in a large containers, and what is that large container is called as fermenter, or it is called bioreactor. Large container, 1000 liters. Large container means how much content it have, means 1000 liters, like that. So that process is called biochemical engineering. So what is the biochemical engineering involves? Biochemical engineering involves nothing but growth of desired microbes. So growth of desired microbes means which microbe? E. coli. So why only E. coli? Because E. coli is only acting as a host. Inside E. coli only our recombinant DNA is present. So that is the reason the microbe which are growing inside the fermenter is E. coli. So that growth of E. coli, okay, that growing of E. coli inside a large container, that large container is called as a fermenter, and applying a technology for growing the microbes in a large container is called as a biochemical engineering, students. Okay, so like that we have to remember. So main techniques, two major techniques that are used in biotechnology is genetic engineering and biochemical engineering. So you have to remember these two terms, students. In multiple choice question they'll ask you what are the two techniques involved in biotechnology. So no doubt it is genetic engineering, and another one is called as a biochemical engineering. So these are the two things which we have to remember here. Okay, so yes, Asmita. So Asmita was telling that oldest application was birth of cover was. Maybe at the time no doubt the fermentation is present in olden days. So at the time also it may be present, no doubt about it. Okay, so let us continue with few more points. So genetic engineering. So finally genetic engineering is a manipulation, no doubt about it. It is a manipulation of genes. It is a manipulation of genes by the man. So no doubt genetic engineering is, no doubt it is a manipulation. Okay, manipulation or modification. So genetic material to alter. So genetic material of DNA is alter the phenotype of the host organisms. Okay, so phenotype of the host organism to suit the human needs. So in genetic engineering what we are doing? We are manipulating, we are modifying the genes artificially in the laboratory. So due to modification of genes, what happens? The organism's basic properties are changed, and new properties were exhibited by the organism, and that new properties exhibited by the organism is suitable for the human needs. Okay, it is helpful for the humans to produce the products which are helpful for human mankind. Okay, so what is this particular sentence? This sentence mean to say like genetic material alter the phenotype of the host and it is suiting the human needs. So this is the sentence. What is the meaning of this sentence is, so due to application of genetic engineering, we are manipulating, we are modifying the genes of the organism. Due to modification of the genes of the organism, what happens? Its normal properties are altered and it gets a new properties. With that property, it is helpful for humans in producing the products which are required for the humans in a day-to-day life. So that's what that particular meaning like phenotype is telling about. Okay, next. So 3 techniques of genetic engineering. So there are 3 different types of techniques of genetic engineering. First one is called as a recombinant DNA technology. Second one is called as a gene cloning. So third one is called as a gene therapy. So just now we were discussing the same thing. So 3 techniques. So 3 techniques of genetic engineering. So this is another important point which we are to remember in multiple choice question. They'll ask you what are the 3 techniques in genetic engineering. So 3 techniques in genetic engineering are recombinant DNA technology, gene cloning or gene amplification. So can anybody tell me how do we do gene amplification, students? How do we do gene amplification? Yes, anybody can answer this question. So how do we do gene amplification? Yes, students, anybody can answer this question. How the amplification of gene will be taking place, by using which technique? By using which technique can we amplify the gene? Yes, students. Yes, anybody, students? Yes, very good, very good, very good, very good. Essay node and very good, Madhurima, very good. So it is nothing but PCR. So by using PCR, what happens is very good, Asmita, very good, Ruba, very good, Vardhan, Tejvardhan also very good. So no doubt it is polymerase chain reaction. Very good, very good, very good. So it is using the PCR technique. No doubt amplification can be done by using a technique. The name of the technique is called as a polymerase chain reaction. Okay. So can you tell me what is the enzyme that is involved in PCR? What is the enzyme that is involved in PCR, students? Can anybody tell me what is the enzyme that is involved in the PCR? So what is the basic enzyme involved in the PCR, students? Anybody? Yes. Can you tell me what is the basic enzyme? Yes, students, basic enzyme. So what is the basic enzyme involved in the PCR? That is my question. Basic enzyme. Yes, very good, essay node, very good, Asmita. So very good, all of you. Yes, very good. So it is nothing but it is the tag polymerase, right? Very good, Komala. So tag polymerase. Okay, so tag polymerase. Very good, students. So very good, Komala, and very good, Saurav. So it is the tag polymerase. So tag polymerase is the, yes, so very good, Rathod also, Rathod. So it is tag polymerase. So tag polymerase is the enzyme. Yes, tag polymerase is the enzyme that is involved in the process of the genetic engineering. Okay, tag polymerase is the process. Okay, so let us define what is recombinant DNA technology. So in recombinant DNA technology, what happened? Hybridization of DNA. Okay, so recombinant DNA technology is hybridization of DNA from different sources. So hybridization of DNA from different sources to achieve the desired genotype and phenotype of a organism. So what is the main aim of recombinant DNA technology? So main aim of recombinant DNA technology is to achieve the desired genotype. So whatever genotype the particular scientists require, they prepare. Okay, they prepare the required genotype by using applications of recombinant DNA technology. So similarly, if they have same type, if they have a desired genotype, certainly the desired phenotype also will be produced. Okay, so the main purpose of recombinant DNA technology is to achieve the desired genotype and as well as phenotype. So recombinant DNA technology is based upon 2 important discoveries. So it is based upon the 2 important discoveries. So discoveries in bacteria. So let us see what are the 2 important discoveries in bacteria. So first one is presence of the plasmids. So presence of the plasmids. So you can see here in bacteria we have plasmids. Okay, so plasmid also undergo replication, no doubt about it. Okay, so the plasmid is one of the important discovery that is related to recombinant DNA technologies, the discovery of plasmid. So plasmids are present only in bacteria. So can anybody tell me what is the nature of the plasmids? Yes, students, can you tell me what is the nature of the plasmids? Yes, what is the nature of the plasmid? How the plasmid appears? Is plasmid DNA or RNA? How it appears? Can anybody tell me what is nature of plasmids? Yes, students, what is the nature of the plasmids? Any answer from you? Yes, so what is the nature of the plasmid? Means how the plasmid appears. That is my question. What is the nature of the plasmid? How they appears? That is my question. So no doubt, very good. So they very good, obviously their independent replication takes place, that is called autonomous replication, no doubt about it. So just now Bhavya was telling the same thing. So it undergoes autonomous, plasmid undergoes autonomous replication, no doubt about it. So it undergoes autonomous replication or self replication. Okay, so that is the right, no doubt about it. Yes, so very good, very good. It is circular. Yes, very good Asmita. So if you see the nature of the plasmid, it is no doubt it is double strand DNA. So it is the dsDNA. dsDNA means it is double strand DNA. It is double strand, circular, and how it appears? It is double strand, circular, and it is closed. So it is double strand, circular, and closed. So this is the nature. Okay, so these are the things which we have to remember in multiple choice questions. They'll ask you like plasmid is. Okay, plasmid is having what type of character feature? So what is the character feature of the plasmid? No doubt they are double strand DNA. Okay, they are circular, double strand, closed. They are circular, closed, double strand DNA. So like that we have the answer actually. Okay, yes. So if you see the plasmid, here, every plasmid have one specific site. Now this is called as a, ori site. It will be having a specific site, that is called as a ori site. Okay, so this ori site is involved in the process of, it is nothing but ori site means origin. It is origin of replication. So ori site is nothing but it is the origin of replications. Yes, very good. So very good students. So very good Bhavyasri, very good Saurabh, they replicate independently. Very good Madhurima, very good Priyanka. So yes, so Bhavyasri, very good. So origin of ori site is nothing but origin of replication. Okay, so this plasmid, so discovery of plasmid made easy for recombinant DNA technology actually. So why? Because plasmid is having that properties. So every plasmid has one specific site, that is called ori site. So that ori site is called origin of replication site, from where replication is initiated, so that autonomous replication will be taking place. And if you see the properties of plasmid, so plasmid is, what is the property of plasmid? Plasmid is a double strand DNA. It is circular, no doubt about it. It is closed. It is circular and closed. You can see clearly it is circular and closed. Can anybody tell me the properties of the bacterial DNA, students? How the bacterial DNA appears? Can anybody answer this question? How the bacterial DNA appears? That is my question. Yes. Yes, very good Saurav. You are also giving good information. Madhurima, Saurav, very good. So no doubt the special physiological characteristics are given to bacteria. No doubt, like in plasmid we have some special physiological characters like the presence of antibiotic resistant genes. On plasmid also we have the antibiotic resistant genes will be present. Very good, no doubt about it. Very good Saurav and Madhurima also. So they also have some enzymes, no doubt about it. Yes, can anybody tell me? My question is, what is the properties of the bacterial DNA? If you see the bacterial DNA, so no doubt bacterial DNA is also double strand. It is also a double strand DNA. Okay, it is also double strand DNA. It is also circular. Okay, it is also circular. It is double strand DNA. It is also circular and it is also closed. Okay, so it is closed. But plasmid is extra chromosomal DNA. Okay, plasmid is a extra chromosomal DNA, whereas bacterial DNA is a bacterial DNA. Okay, it is a part of the, it is having this bacteria, it is nothing but it is only single chromosome is present actually. Okay, so in bacteria we have only single chromosome. On that single chromosome we have the bacterial DNA, and the nature of the bacterial DNA is double strand DNA. It is circular and closed. Okay, so like that we have to remember, students. So very good. So welcome to Kanyakumari. So welcome to Unacademy EAMCET YouTube channel, students. So once again, so what is the important thing which we have to remember here is, what is the nature of the bacterial DNA? If you see the nature of the bacterial DNA, no doubt bacterial DNA is also double strand DNA. It is circular, it is closed, and this bacterial DNA actually, it is the nucleus of the bacteria is called as a nucleoid. Okay, it is called nucleoid. This nucleoid has only single chromosome. On that chromosome we have DNA, and what type of DNA is present? It is double strand, it is circular and closed. Similarly, there is another DNA is present, that another DNA is called as a plasmid DNA. So that is the reason it is called, plasmid is called as extra chromosomal DNA. If you see the nature of the plasmid, it is also a double strand DNA, it is also circular and it is closed. So these are some properties of the chromosomal DNA, okay, and plasmids. Okay, students. So finally what we have to remember here is the discovery of the plasmid is a very good finding in case of the recombinant DNA technology. Okay, next. So recombinant DNA is also based upon two important discoveries. So just now we have seen that the discovery of the plasmids. After discovery of plasmids, the discovery of restriction endonucleases. So there is a discovery of restriction endonuclease took place. So there are nearly, so how many enzymes has been identified till now? So till now nearly 900 restriction endonucleases were identified till now. So more than, nearly or more than actually. So 900 restriction enzymes has been identified. So rDNA technology, so two important discoveries. First important discovery is discovery of plasmid, and second important discovery in recombinant DNA technology is discovery of restriction endonucleases. So what is the source of restriction endonuclease? That is my one question, students. Can anybody answer this question? What is the source of endonucleases? Anybody, students? What is source of these endonucleases? Anybody can answer this question? What is source of endonucleases, students? What is source? What is the source of restriction endonucleases? Yes, anybody? Any answer, students? Yes, very good, Saurabh. So no doubt more than 900 restriction enzymes has been identified, and many strains are involved, not only 230 strains, several strains. Till now the number of restriction endonuclease identified nowadays is in lakhs only actually. But what happens is, this number 900 was of 1985. In 1985 this number came, like 900 restriction enzymes has been identified. By today it was in number of restriction enzymes has crossed the lakhs also. Okay, so anyhow, what is there in textbook, that only we have to discuss and write in the exam. So based on that we will remember that. So nearly 900 restriction enzymes has been identified. Now my question is, what is the source of endonucleases? What are the main sources? Yes, very good. So very good, Sunny, and very good, Asmita. The only one and one only source. Okay, one and one only source of restriction enzymes is nothing but it is bacteria. So bacteria is the only one and one source. So restriction enzymes can be identified only from bacteria. Only restriction enzymes can be identified, students. Restriction enzymes cannot be extracted from virus. They cannot be extracted from protozoans or any other organisms. Restriction enzymes, the only one and one solo source. ఒకే ఒక్క సోర్స్ ఏదంటే బ్యాక్టీరియా. సో బ్యాక్టీరియా నుంచే రిస్ట్రిక్షన్ ఎండోన్యూక్లియస్ అనేది ఎక్స్ట్రాక్షన్ జరుగుతుంది. ఓకే, సో రిస్ట్రిక్షన్ ఎండోన్యూక్లియస్ is also called as a molecular scissors. Okay, or it is also called as a biological scissors. So restriction endonuclease ki another name, it is called as a biological scissors or it is called as a molecular scissors. So why it is called biological scissors or molecular scissors is because it cuts the DNA. As it cuts the DNA, it is called as a molecular scissors, or it is called as a biological scissors. Okay students, so we'll stop here. We'll continue the another part in the next session after 2 days. Again my class will be there, so after 2 days you join students, so we will discuss, and you can join our special live class or Unacademy app also. You subscribe our Unacademy EAMCET YouTube channel so that you can know the timings of our classes. Okay students, thank you all of you. Thank you for attending. See you in the next class. Thank you students. Bye bye all of you.